Good morning, everyone. Today we are going to take the third module of molecular basis of inheritance. In this, we are going to deal with the DNA replication. So, in the last module, we have discussed about the experimental proof for the semi-conservative mode of replication. So, here the DNA replication, it is the autocatalytic function of DNA. So what do you mean by autocatalytic function means? The DNA molecule itself is taking initiative for the synthesis of its own type. Hence it is called as autocatalytic. Then it follows the semi-conservative mode of replication. So that means since the DNA is double-stranded, both the strand will act as the template. And based on the base sequences, the complementary base pairing will take place. And as a result, two new daughter strands will be formed from a single DNA strand. In that, each new strand will get one old template strand as well as one new strand. Here, the old strand is conserved. One of the old strand is conserved in the new molecule. Hence, it is called as semi-conservative mode of replication. Then, when the replication is taking place, so it is at the time of cell division. And in the cell division itself, in the phase S phase, synthetic phase, where only the replication of DNA is taking place. And in the DNA itself, which location the DNA replication is starting. So here there is a special sequences located, and that sequence will give the signal for replication. So that area where the DNA replication takes place is called as ORI, origin of replication. So there will be 35 sequences before the ORI. There will be repetitive sequences. So the cell machinery will recognize that after 35 sequences or 10 base sequences, the origin of replication or ORI will be present. So in prokaryotes, since they have single circular naked DNA, there is only one ORI. Even in virus also there is only one ORI. So there the origin, the DNA replication starts, and in most of the prokaryotes, that is the direction of replication is bidirectional. That means one strand towards the 5'3" direction, the other strand also in the opposite direction. So both strands, that is from the point of origin, one strand will move this way, means the other side it will move this way. So bidirectional. That is why the replication time for the E. coli or DNA replication time for the E. coli, it is one replication, it requires only 18 minutes. Only 18 minutes. But when you are taking the DNA, the base sequence of the entire DNA, the speed when we are considering, it requires 38 minutes. 38 minutes. Since here in each time from the array, both direction, the elongation or the replication starts, it requires only half the time, that is approximately 18 minutes. The remaining 2 minutes is for cytoplasmic division. So the DNA division in E. coli, that is actually 38, but here it is bidirectional, so half the time only they require. That is why 18 minutes the DNA replication is finishing. So that means it is bidirectional. It is bidirectional. Now we will move on to the steps. When you are saying the steps here, I have just given how the semi-conservative mode of replication is taking place. Just a diagrammatic sketch. So here both the strands will retain in the new strand. Okay. So hence it is called as DNA semi-conservative mode of replication. Then we will move on to the next concept. So how this DNA replication is taking place. That is with the help of enzymes. So the main enzyme concerned in that is DNA dependent DNA polymerase. DNA dependent DNA polymerase. There are three types of DNA dependent DNA polymerase. DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. So their respective roles we will deal it in the next session. So here already I said there is a particular site in that. So in eukaryote, that site is called as ore, where the replication is taking place. And in eukaryotes means it is linear DNA. It is not circular DNA. The DNA is linear. That means a rod-like structure. So they have open ends. Both the sides they have open ends. But the length of the DNA it is very high. In human beings means it can reach up to 2.2. So from one end to the till the last end, if it is opening, then it will lead to more complications. So instead of that, eukaryotes, the DNA replication takes place in many places. That means nearly 1,000 ori can be seen. Okay, in eukaryotic cell, if it is in bacteria, it is only one ore and it is bidirectional, means in eukaryotes, it is unidirectional, and the number of ore, many places we can see, origin of replication, and finally all these places unite together, and from that the two independent DNA will be produced. So the ore, the number of ore, many ore can be seen, and nearly 1,000 ore will be there in the human DNA. Then we will move on to the next one. So what is the relevance of this array? Because they have a special sequence, the cellular machinery, that means the DNA, DNA replicating enzymes, and the factors, proteinaceous factors will bind onto that particular site. And they results in unwinding and unzipping of the base sequences. That means the bond between hydrogen bond between the base pairs will break. So that results in two single-stranded DNAs. And both the single-stranded DNA region will act as the template for the new synthesis. So as a result, when they open up at the ORI region, we will get a Y-shaped open region. That region we can call it as replication fork, and that is mediated by and catalyzed by the DNA polymerase enzyme. So DNA polymerase enzyme is essential. Along with that, because the building blocks of DNA itself is nucleotides, we require all the different types of nucleotides, that is ATP, GTP, CTP and TTP. And here this nucleotides perform dual function. So the first function itself, they are acting as the substrate, because the DNA itself is polynucleotide, and in that the nucleotide itself is either ATP or GTP or TTP or CTP. So that is acting as the building block or the structure or the substrate. And apart from that, this polymerization reaction, phosphodiester bond formation, requires the energy. So that it is acting as, apart from this nucleotides, apart from acting as the substrate, they are also providing energy for the polymerization, or we can say the formation of phosphodiester bond between the neighboring nucleotides. So it is providing energy, and it is also providing the substrate for the polymerization of new synthesis of DNA. Hence, the DNA replication process as such, we can call it as the energy expensive process. And it is mediated by the magnesium ions also. For the synthesis of DNA, we require magnesium ions. And here I will explain what do you mean by the replication fork. So suppose in that both the direction, always the elongation or the formation of new strand will be always in the 5 to 3. That means the direction of template strand will be always 3 to 5 direction. So in the template strand, one of the strand there will be a small nick formation or a small break will take place. So as a result of that nick formation, That means in one strand, the phosphodiester bond at a particular region, they will break. So that itself will relieve the tension and it will help in opening up. So that is done by unwinding of the DNA helix is done by the enzyme that is called as helicase. Helicase helps in unwinding the DNA helix. But even then, for a long time, the DNA is in the supercoiled manner and it is encapsulated within the nucleus. They will have a recoiling tendency. So in order to relieve the tension of recoiling tendency at the ori, one more enzyme will bind. That enzyme is called as topoisomerase. So with the help of helicase and topoisomerase, which will act on the ori, and there results in unwinding of the DNA helix. So when the DNA helix in a particular region starts unwinding, we will get a Y-shaped opened area. So that Y-shaped opened area is called as which one? The replication fork. Now we'll move on to the next slide, that is which all enzymes are actually taking part in the DNA replication. So the first and foremost enzyme when we quote, it is none other than the DNA dependent DNA polymerase enzyme. And here already I said 3 types of DNA polymerases. DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. And in eukaryotes it is even more complicated. So here DNA polymerase 1, it is for proofreading and it is for proofreading and primer removal. DNA polymerase 1 is for proofreading and for primer removal. And DNA polymerase 2 is for DNA repair. And the DNA polymerase 3 is actually for the polymerization of deoxyribonucleotides. So polymerase 1 is for proofreading and primer removal. 2 is for repair. If any error is there, means that will be removed and then it will be repaired. And DNA polymerase 3 is for polymerization of the free nucleotides, and then finally as a result we will get a new polynucleotide strand. And as I already said, the helicase. Helicase is the enzyme which helps in unzipping the DNA strand. How the DNA strands are unzipping? By the breaking the hydrogen bonds between them. Thus it helps in the formation of replication fork. And since this unwinding results in recoiling tendency, one more enzyme is there that is called as topoisomerase. In prokaryotes means we will say the same enzyme with another name that is called as gyrase. So this enzyme will prevent the recoiling tendency of the double-stranded DNA. Then there is another enzyme that is called as primase. So here primase means the DNA polymerase enzyme, that is especially DNA polymerase 3, DNA polymerase enzyme as such, it cannot initiate the DNA polymerization. That means it does not have 5'3" endonuclease activity. 5'3" endonuclease activity. That means 5 direction to 3 direction, that phosphodiester formation. That formation DNA polymerase is not having because the new synthesized strand is always in 5'3" direction. So they cannot initiate the polymerization. Once there is a third free end, 5 to 3, 3 end is there, means they can establish. Once if there is an end, then to that they can start, they can elongate, since they do not have the 5'3" exonuclease activity. So for that, how the cell is managing that means they will form a primer. So primer means it is a small segment of ribonucleotides, which can be of up to 12 base sequences, and that bases are of ribonucleotides. A small strand of ribonucleotides, that is oligonucleotides, we can say, that is can be of maximum less than 10. So 12 we'll say, but even then. It's a single-stranded oligonucleotide strand made up of ribonucleotides, which will bind at the third end of the template, which will bind and base pair and establish a base pairing between the old strand, third end of the old strand, and the new strand. And that binding is taking place with the help of an enzyme. That enzyme is called as primase. So here I will read. This enzyme helps in the synthesis of RNA primer complementary to the DNA template strand. And once that small end of with the primer is taken place, means that step is called as initiation. So once the priming is done, then there is a free terminal, and with that the DNA polymerase can elongate. With the help of DNA polymerase, the new strand can be elongated by establishing the polynucleotide, by establishing the phosphodiester bond. So since DNA polymerase cannot initiate the DNA polymerization in the 5'3" direction, we require a small stretch of single-stranded oligonucleotide made up of ribonucleotides, which will be attached at the third end, or which will form base pair with the third end of the DNA template strand, which is mediated by the primase enzyme. There the initiation will take place. Once the initiation has taken place, then elongation is done with the help of DNA polymerase. Then there is one more enzyme that is called as ligase. The role of ligase, wherever there is any cut ends, so that cut ends can be joined with the help of DNA ligase. It is acting as a glue. So in the DNA itself, when the DNA strands are forming, one strand will form continuously, the other strand will form discontinuously, because the polarity of both the strands are anti-parallel. One strand is 5'3" direction, means the other template strand is 3'5" direction. So always the new strand synthesis is in the direction of 5'3". Then the template which is having 5'3" direction, there the polymerization cannot take place in a continuous manner. So there the formation is in the form of small stretches of DNA polynucleotides, and that stretches are called as Okazaki fragments. So this Okazaki fragments are united with the help of DNA ligase. So ligase is helping in gluing the discontinuous DNA strands, and they conceal, or they can seal the gaps, and they can reunite the cut ends. Then there is, apart from that, there is endonuclease. They produce a single-stranded or a double-stranded cut into a DNA molecule. So wherever there is cut, they can unite it, or they can act on. Then apart from that, there is one more proteinaceous factors that is called as single-stranded binding proteins. So as I already said, there is an ori. At the ori, there is a nick formation, there is a cut formation. As a result, the tension, when the helix and helicase and the topoisomerase is binding, there will be a tendency of recoiling together, even though topoisomerase is helping to relieve the tension, but the stability of the single-stranded condition, because for a long time this coiling is taking place. So recoiling tendency they will have. So in order to avoid the recoiling, both the strands will act like single-stranded for a long time. So in that bend region, in that fork region, replication fork region, the single-stranded binding proteins will bind. So they will give them stability to remain as single-strand itself when the replication is going on. So the purpose of single-stranded binding protein is to stabilize the single-stranded condition when the replication takes place. Now we will move on to the next one. The first step already I said, initiation. So initiation is nothing but unwinding of the DNA with the help of helicase and topoisomerase at the ori. And as a result of breaking, unzipping of the nitrogen base pairs, the nucleotide base pairs, we will get a replication fork. And at the template region, so both the strands will act as the template. So the strand, DNA template strand with 3-5 direction, there the attachment in both the sides, it is actually the attachment of primer, and that will initiate the DNA replication. So once the initiation taken place, so here in template strand with 3-5 direction, continuously it can proceed. Once it is started, it can continuously proceed. But simultaneously on the other template, where the direction of the template strand is 5-3, always the newly synthesized strand, the direction is 5-3. Since both the DNA strands have to be maintained anti-parallel, the primer will attach little more far away from there where the third end is present. So at the third end of the template, a particular region away from that, from there the primer attachment, and in the reverse direction the initiation and elongation takes place. So in that way, two region, so one region continuously 5-3, and the other region it is cannot be formed because that end, it is one end it is open, the other end is coiled condition. So there it has to wait for the uncoiling of that region. So there it is, the elongation is discontinuous manner. The template strand, which is having the direction of 5-3, template strand, the direction is 5-3. There also the DNA replication is always taking place in 5-3 in opposite direction, and it is in a discontinuous manner. Soon after the primer attachment, with the help of DNA polymerase, the new nucleotides will be added complementary to the template. So one strand it is taking place, elongation is in continuous, that strand is called as leading strand. And the other strand, it is discontinuous manner because the template strand itself is, the direction is 5-3. Always from third end to the 5, and again third end to the 5. In that way, small fragment manner only it can be produced. So that individual fragments, each fragment again they have to initiate with a primer. So many primer is required for the lagging strand. And the direction of formation in Okazaki fragment formation is always 5-3 direction. But here and there gaps will be there. So in a discontinuous manner, the elongation in opposite direction, it is taking place. And the Okazaki fragments, when you are saying it is approximately 200 base pairs. And each Okazaki fragment, the direction is 5-3. And they starts with, which one starts with a primer. So that process is called as, it is mediated elongation, means the establishment of phosphodiester bond in 5-3 direction. So this elongation also is mediated with the help of DNA polymerase 3. DNA polymerase 3 enzyme. Now when it reaches, so in prokaryotes, since it is unidirectional, it will bidirectional, both ends will finally will reach up to a particular point. But in eukaryotes, there are many ori and many replication fork. Everywhere this elongation bubble, replication bubble, and there the elongation and the replication is taking place. Finally, ultimately, so wherever this the DNA strands are formed, soon after that, which one will take place? That is, once the two strands are formed, immediately, wherever they are, two strands are formed and that completed. So before completing, there is a termination process. That means the termination process, we cannot say literally in that particular point everything is coming to an end. It is not like that. Wherever the replication fork is forming, replication bubble is forming. So from one ori to the next region. So when it reaches the next region, automatically the proofreading and repair will take place. So proofreading means, proofreading and repair is proofreading and primer elimination, primer removal. Proofreading is done by the DNA polymerase 1. So which is having both the activities, 5'3" activities and 3'5" activities. Hence, they will roll in a reverse direction. So they will check whether as a result of elongation or as a result of replication, all the base pairs are correctly matched or not. And if any particular region, if any wrong base pair happens, means with the help of repair enzyme, that is DNA polymerase 2, it will excise that wrongly paired base and insert a correct pair. So that is called as proofreading and repair mechanism. So as soon as the proofreading and repair mechanism has finished, then the helix reformation will take place. So many places the replication at a time it is taking place in eukaryotes. So here we will move on to the slide, means the Okazaki fragments. They are the small stretches of deoxyribonucleotides which is having the direction 5' to 3', and each fragment consists of approximately of 100 to 200 base pairs. Bases starts with primer, and that is it is formed in the lagging strand, and whose template direction is 5'3". Okay, 5'3". So in later, the Okazaki fragments, the cut ends of the gaps between the Okazaki fragments are inserted, or there the primer will be removed, primer removal, DNA polymerase 1, and the correct deoxyribonucleotides, the correct matched opposite one, will be fixed or will be inserted, and that is done by the DNA polymerase 1. So primer removal and the correctly base pairing is done. Then these cut ends are ligated with the help of ligase. So in lagging strand we need many primer, and at the same time the ligase molecules also need compared to the compared to the lagging strand. And here one more point we have to mention: a failure of cell division after DNA replication. So DNA is replicated, but instead of after that karyokinesis, if the cytokinesis is not taking place, so within a single cell itself, the number of DNA will keep on increasing. That means the number of chromosomes will increase. So that condition is called as polyploidy. Then the error rate, when I am saying the DNA, the error rate compared to RNA formation, the DNA error formation is very, very less. That is 1 in every 10 to the power of 4 to 5 base pairs. One error can take place, because the maximum the DNA polymerase will try to remove the error. Repair mechanism, with the help of proofreading and repair mechanism, it can remove the error. So error chances, that is why when we say the chance of or stability of DNA is more compared to RNA. Now we'll move on to the next one, termination. Already mentioned only termination. So termination of replication occurs at different ways in different organisms. In E. coli, that means in prokaryotes, the chromosome is circular. And this happens when the two replication forks between the two terminals meet each other. But in eukaryotes, since it is linear, many places the replication starts with the ori and it will proceed along the replication fork. So many places, so as soon as the elongation takes place, simultaneously the proofreading, then primer removal and all will take place, and helix reformation will take place. Then DNA polymerase adds nucleotides on one of the template strand, which is called as leading strand, which is having the template polarity 3' to 5'. In this strand, nucleotides are added continuously, therefore it is called as continuous replication, and that strand is called as leading strand. So on the other hand, the replication is discontinuous. Small fragments of Okazaki formed, small fragments of nucleotides are formed and are called as Okazaki fragments, which are later joined by DNA ligase. That strand is called as lagging strand. Lagging strands template, the polarity will be 5 to 3. Then the main summarizing the main steps in DNA replication in prokaryotes. The DNA replication in prokaryotes takes place in the following steps. The first step is the two strands of the DNA unwind at the origin of replication. Then helix, with the help of topoisomerase, opens the DNA and the replication fork is formed. So helicase is for unwinding, means topoisomerase is for relieving the tension. And as a result, we'll get two single-stranded DNA templates. Then the DNA is coated by single-stranded binding proteins around the replication fork to prevent the rewinding of DNA. Topoisomerase prevents the supercoiling of DNA. Then RNA primers are synthesized by the primase enzyme, and these primers are complementary to the DNA strand. And this primer will attach at the 5 end of the template strand. And the primer as such, when we are saying they are the ribonucleotides, which consists of approximately 4 to 12 bases. And they are coming under ribonucleotides, and which will establish the complementary base pairing at the 5 end of the DNA. Then DNA polymerase 3 starts adding nucleotides at the end of the primer. Then as a result, one strand, the replication is continuous, and in the other strand, it is discontinuous. So which is taking place continuous in 5-3 direction without any interruption. That strand is called as leading strand, where the polarity of the DNA strand is 3-5. And there we require only one primer. Whereas in the other strand, where the polarity of the template strand is 5-3, there continuous synthesis is not possible, so it is formed with the help of Okazaki fragments. Each Okazaki fragment starts with primer. And as a result, leading strand and lagging strand will be formed. Then at the end of the replication, the primers will be removed and the gaps are filled with DNA polymerase, and the cut ends are joined with the help of DNA ligase. So as a result, we will get two DNA molecules from a single DNA strand. And as soon as they form the two strands, the helix reformation takes place. So the entire process will come to an end. So with this, I am concluding the DNA replication. We will meet again in the module 4. Thank you very much.